Hey everyone, this is Elan, CEO and co-founder of Astrolabe Diagnostics, and today I'd like to talk about one of the ways in which we benchmark Astrolabe's cell subset labeling. Earlier this year, we published this paper with Adib Rahman of Mount Sinai, and here we're showing how to use Astrolabe as part of a standardized pipeline for acquiring immune monitoring data using the CyTOF. And the paper touches on a few of the techniques we use for validating the data. And specifically, we talk about the Matthews correlation coefficient. And if you go to figure 3, then you will see that we calculated that statistic for each one of the different cell subsets. So this is one way you can do this. You can take a manually labeled data set and see how well Astrolabe conforms to these subsets. And again, you can go to the Frontiers paper and see that we have a pretty strong match there. But I would like to talk about a different approach. And what we've done there is we took some of the data from this experiment, and then we've done something very, very simple. We loaded it in Flojo, and we gated it. First of all, we found the siglets through a very basic primitive singlets gate, and then within the singlets, we took just the CD3 positive cells, and then the CD3 negative cells. So we took all of these samples, we split them into CD3 positive, CD3 negative, and then we build a synthetic dataset, which we call the T cell frequency titration dataset. A very fancy name to say that we have 12 samples, and in one of them, 20% of cells are T cells, in the other is 40%, 60%, 80%, and so on. So we build this synthetic dataset with the goal of seeing whether Astrolabe or any other method for identifying cell subsets is going to catch that structure. So after analyzing it with Astrolabe, I'm going to switch to the cell subset navigator. This is a composite view of the entire experiment. Everything I'm showing you here is using data from all of the FCS files, whether the experiment has 10 of them or 1,000 or 10,000 files. And I'm going to turn on a tool called differential abundance analysis. So the differential abundance analysis, as its name implies, looks to see whether any of the cell subsets differ in abundance between the experimental conditions. And right now the experimental condition are the T cell frequency that we artificially introduced. So if I go down, Astrolabe is giving me a volcano plot where each of these cells is one of the subsets in the experiment, and the x-axis is the fold change across the different samples, and the y-axis is a measure of statistical significance. And this volcano plot is a bit weird. We see one big streak to the right of T cells and one big streak to the left of non-T cells. And let's pick the naive CD4 T cells and the naive B cells as an example. And when I scroll down, I see that indeed the level of the naive CD4 T cells increase between the titrated experiments, and the level of the naive B cells decreases. So that's a nice positive result. It is what we expected. However, there is heterogeneity within the T cell compartment and within the other compartments, which is reflected here and makes the comparison a bit more complicated. So I'm actually going to flip to a different cell subset level. What I'm showing you here is the assignment level, and it includes a whole bunch of different subsets. This goes as deep as Astrolabe can go using the markers in the panel. The other level is called compartment, and when I switch to the compartment level, we're going to see very few bubbles, and these correspond just to the major compartments: NK cells and myeloids and B cells and T cells and so on. And here, if I pick the T cells, then I'm going to see exactly the frequencies I was looking for. So 20% at 20%, 40%, 40%, 60, and 80%. So again, this is a toy example, and this is something that is very easy for the platform to do. With that said, we do use these benchmarks on a regular basis to make sure that Astrolabe gives researchers the kind of answers that they're looking for. So if you have any questions, please reach me at elad@astrolabediagnostics.com. You can find our website and our YouTube channel. Thank you for your time and have a wonderful day.